Welcome everyone to our monthly seminar series. Thank you so much for those who have been able to join in person so far, and welcome to those who will be watching this after the fact once it is posted. My name is Jonathan Plett. I am the president of the AMS, and it is my very big pleasure to welcome this month's speaker, who is Bernard Slippers. Now, Bernard and I met a number of years ago when we were at a conference in Singapore. I then had the opportunity to go down and visit him in South Africa and the institute where he is working, where they do some really phenomenal work. Now, Bernard has a huge list of accomplishments. He's the director of the Forestry and Agricultural Biotechnology Institute, or FABI. He's the founding director of Future Africa, which was something that was just being built up actually when I was visiting. So I'm curious to hear more about that if you're talking about that at all. He's also a professor in the Department of Biochemistry, Genetics, and Microbiology at the University of Pretoria. So already you can begin to see Bernard is a very busy man. He leads the Tree Protection Cooperative program, and his research there is focusing on the ecology and evolutions of insects together with microorganisms and to see how they affect plant health. Just a final couple of points on him. He is a Clavariate highly cited researcher, and he was elected and a fellow of the Academy of Science of South Africa. And so it's a huge pleasure, as I said. Bernard, thank you so much for talking to us today, and I'll pass it over to you.
Jonathan, thank you so much for the invitation. Can I check that you hear me clearly? The sound is working fine.
Great. Yeah.
Great. Thank you so much for the opportunity, and for those that are joining or watching this later, listening to me talk about a symbiosis that has fascinated me for more than 20 years. The symbiosis between the Sirex noctilio woodwasp, which is what you see on the left-hand side here, and its associated fungus, Amylostereum areolatum. And I will be reflecting on the ecology and evolution of this symbiosis in general, also beyond these two species, but then bringing it home to how that understanding of the ecology is relevant to the biological control of the system. It's hard to not be fascinated by this organism. It's a very big wasp. The wasp that you see on the left-hand side there might be 2 to 3 centimeters in length. And you see between those orange legs a needle-like ovipositor that she's busy drilling through the bark of a pine tree. Do I advance? Yeah, there we go. So a little bit of background on the symbiosis. You've just seen a photograph of this female wood wasp that's busy drilling through the bark of a pine tree. Those pine trees, she has the ability to assess levels of stress and water pressure within those trees. And if that is too high, so the tree is still healthy enough, she will not lay her eggs. She will make one drill in there, and she will deposit a phytotoxic mucus that will further stress the tree and the fungus, but she will not deposit her eggs. If the tree is susceptible or deemed to be susceptible, she will make more than one drill and also introduce her eggs. Those eggs will develop into larvae, and those larvae in the southern hemisphere at least will spend about 1 year tunneling through the wood. In the northern hemisphere, it might be 2 years or even longer. And I'll say something now about the nutrition within that system. And after a year, these wasps will emerge and fly off to find the next stress tree. An interesting part of the biology that I will stress throughout is the fact that these female wasps then pick up an asexual mycelial fragments of this fungus that she carries in an internal mycangium. But we do know that the fungus, not in our environments in the southern hemisphere, but in its native environment in Europe, can sporulate on the tree and produce sexual spores as well. But that, as far as we know, have known historically, was not linked to this cycle. Oops, I'm going the wrong direction. Maybe just something about nutrition and the role of the fungus. It was long thought that as the wasp tunnels through the wood, that it feeds on the fungus. But really convincing work by Brian Thompson and colleagues some years ago showed that it doesn't actually feed on the wood, and neither does it ingest any, it doesn't feed on the fungus itself, or doesn't derive significant amounts of nutrition from the fungus. And it doesn't actually ingest the wood itself either. Rather, the wood is digested externally by the fungus, and cellulose, of course, being a key carbon resource for this developing larvae. The larvae will scrape off minute pieces of wood from the wall of its tunnel, squeeze out the liquid from that, and ingest the liquid. And from that liquid, the larvae is able to do the final steps of digestion of the cellulose that the fungus had started externally in the wood. I'll point out and not spend too much time talking about this particular or this part of the topic today, but the genome of this fungus has recently been sequenced. We've also sequenced the genome of the wasp, but that is not published yet. This is not our sequence, but Fu and colleagues, a 53 megabase pair, and I see there's a typo there, genome that is enriched for cazymes, as we would expect for one of these wood rotters, but seems to be have reduced virulence related genes, and the authors speculate that this is what we would expect for an organism like this that is introduced into a suitable host by a suitably stressed host by its associated wasp, but that really needs this cazyme repertoire to digest the wood for itself, but also for its associated insects. So just to give you a sense of what that looks like in real life. So here are some images and a small video giving you a sense of the effort that it takes for that female wasp, this is a Urosphorus species, to drill that needle-like ovipositor through the bark to ultimately deposit her eggs underneath the bark. If you dissect that wasp's abdomen, you will find a large mycotoxin. So this is where the toxin is produced, this phytotoxic mucus, and these two mycangia, these sac-like structures that are filled with a clone of this particular fungus. When you isolate from this, no other organisms will grow out of it. So a sterile environment, really intriguing exactly how that happens. And you see trees dying here on the right-hand side, and you see the tunneling through the wood here, and these round exit holes at the end of the life cycle. So I've referred to the fact that these female wasps pick up asexual fragments of the mycelium that grows inside the mycangium of this wasp, and that it then transmits to the next tree. But that means that it transmits a clone of the wasp that or the fungus that was in the wood that she developed in. And some years ago, and this I was doing a master's, or at the time, or starting to do a master's degree, colleagues in Europe showed that you could find very large vegetative compatibility groups. So for those of you who might remember this technique that we used to use quite a bit more than today in the past, where you would pair two different individuals on a plate, and if they were incompatible with each other, they would have this antagonistic reaction. But if they were compatible and of the same genotype, the mycelia would fuse. And they could show that you could find these clonal, these clones spread across large geographic distances. Of course, that's what you would expect. These wasps can fly 50 kilometers or more per year, and they would then spread this clone to other areas. So I'm jumping a little bit in the topic to take this technique to our understanding of how this wasp has spread globally. So I've mentioned the fact that it's native to Europe, but it has been introduced to many parts around the world in wood and wood packaging material as we move it around the world. And that gives you a sense of where it has been introduced. New Zealand and Australia was also colonized by this more than 100 years ago now, in South America and pine plantations here in South Africa. More recently been introduced into China, and it's also been introduced into the US. In these introduced environments, unlike in the European environment, we can get large population outbreaks. The video at the top here gives you a sense of a male swarm that's forming around cut wood. This is not something that you would easily see in the European population because the densities are so low. But in our South African environment, when there are large outbreaks, that is something that you could see. And at the bottom of the slide, you see the extent of the death that this could cause in these pine plantations. So a very serious pest in these invaded regions. So we've been interested in the origin of these invasions and have used this fact of the clonal spread of the fungus by the wasp to study that spread. And what you see here is work already now published quite some years ago, where we looked at populations from South Africa and from Brazil. And here I've paired the South African strains, and we at that stage only saw one clone in the whole South Africa, one clone in the whole Brazil. And when we paired those with each other, they also fused. And some of the molecular markers we used at the time supported that view that us and South America shared an origin of this invasion. It most likely spread from South America to South Africa. It was no longer in South America, of course. So you could use this feature to track the spread of this. So when we later years acquired a population of the wasp itself to study its population genetics, this is what we expected to see. A European origin, spread to New Zealand, eventually to Australia, from there to South America, and from there to South Africa. So we had a large number of specimens, a good collection of or set of microsatellite loci, and the COX-1 locus that we could sequence to look at the diversity. And what we found from that completely surprised us. Not a simple introduction pathway, but rather it looked like many populations, such as South Africa, there had been multiple introductions over time from different origins, so potentially even from Australia, New Zealand, and South America into South Africa. Similarly, a North American population seeming to carry alleles that represent different geographic origins. And also you see in the top right-hand corner, some admixture then between these populations, between these invasive populations. And in fact, the population also suggested that there was a population that we hadn't sampled within our collection that also contributed to the genetics within these invasive populations. So if you summarize that, the yellow part on this particular image, these yellow arrows, yellow and blue arrows, indicate the invasion pathways that we could deduce from the population genetics of the wasp. But that contrasted somewhat with the diversity that we saw at that stage in these Amylostereum areolatum populations. So I talked about this extensive clonality in South America. So it was a little bit hard to explain how we could deal with a clonal population of the fungus, yet there seemed to have been introductions of the wasp from more than one location. This puzzle was solved for us some years ago, or some years later, when we looked at populations of the fungus associated with wasps in invaded areas in North America. So Sirex noctilio is the invasive wasp there. It's expected to carry Amylostereum areolatum. And in those populations, we, with sequence markers there, characterized 3 different haplotypes. And Sirex nigricornis, a native wood wasp that's expected to carry another species called Amylostereum calleti. But if I put on the colors here, and Sirex noctilio is colored in orange, Sirex nigricornis in green, you would see that Sirex noctilio carried, that these Amylostereum areolatum genotypes were carried by Sirex noctilio, of course, but also by Sirex nigricornis. And in fact, even some of the Sirex nigricornis wasps carried some Amylostereum areolatum isolates. So what this suggested was that after invasion, there had been exchange of these species even between Sirex wood wasp species, and that's something that wasn't known to occur before. There was some concern about that because there's a nematode biocontrol agent that I'll talk about later that depends on feeding on this fungus, and that potentially could be influenced by this exchange of the fungi. There's been a number of studies that have subsequently shown the same thing, that Sirex nibricornis, for example, could carry both Amylostereum areolatum and Amylostereum calleti. So apparently not an anomaly that we observed, but something that happens quite frequent in these invaded regions. And even in areas where the Sirex noctilio was not yet known, they could observe some of this exchange that it must have happened historically. We've done work in Japan. Similarly, there I've studied two different wasp species, two different fungi, and could show that these fungi were exchanged not only between Sirex species, but even between Sirex and Uroceras species. So here, Amylostereum calleti, that's supposed to be the associate of Uroceras, which is colored in blue here, but also being carried here by a Sirex species that is colored in red. So this exchange of these fungi happening in these invaded regions, we also, this is a very old slide of work that I did as a postdoc in Europe, looking at diversity of Amylostereum calleti and showing that different Uroceras species in Europe would carry the same genotype. So clearly between those Uroceras species, there is exchange. But curiously, if you look at the color of the dots on the map itself, this particular genotype, a dominant genotype, there's a sequence data, there was spread across Europe and North America. So is this fungus, which is thought to be native in both of those areas, or wasn't thought to be introduced in either of those areas, perhaps also introduced and then exchanged between invasive and native wasps? And the answer is indeed, we see quite a lot of that. So in recent work done by Catherine Fitzer, together with colleague Jeremy Allison, she has looked at a stored collection of Siricid woodwasps from North America and have characterized the fungi in their mycangial sacs from these stored wasps. So she extracted that from the stored wasp, amplified these barcoding markers, and if the red color here indicates the sequences that she's added in amongst the reference sequences. And I know it's small, but a number of Uroceras species here carrying this Amylostereum calleti, Sirex nibricornis, other Sirex species, other Uroceras species. So quite wide diversity of these both Uroceras and Sirex species that must have historically exchanged these Amylostereum calleti isolates, but also species such as Amylostereum areolatum, that's indicated here, both Sirex nitidus, Sirex dianeus, Sirex noctilio, and other wasps. So if we go into literature and from these sequence data that we've produced and look at the associations of these wasps, which have historically been thought to be very, strictly vertically transmitted. So one would expect a strict coevolution between these Amylostereum species and the Siricid species. You see that this associations does not reflect that strict coevolution between these different groups. So something like Amylostereum areolatum being used by a number of Sirex species, but also a number of Uroceras species that we could identify it from. These red lines just indicating new associations we could show. And Amylostereum calleti being even more promiscuous, being carried by many different Sirex and Uroceras species. So clearly not a strict pattern of coevolution, and perhaps we should expect that just looking at the level of diversity amongst Amylostereum and the Siricid species. The last thing perhaps to highlight here is a new species of Amylostereum that we discovered as part of this work, associated also with both Uroceras and Siricid species in North America. We're doing very preliminary molecular dating work, and this pattern, I think, is also reflected there. So the Siricid, the main groups appear to be diverging about 50 million years ago. We don't see any clustering of diversification amongst these Siricid species related to geographic area. North America and Asia containing bulk of the diversity, not all the Asian species reflected here, but clearly these wasps have also more recently than 50 million years ago colonized between these two different regions, and there are known pathways through which that could happen. That is in contrast to the diversity and diversification of Amylostereum species, which appears to be much more recent than the Siricid species. And if we reflect on the fact that these Siricid species exchange these fungi with each other, both at short ecological timescales, but also longer evolutionary timescales, then this is perhaps not so surprising. What it does suggest, though, is that the current diversity of Amylostereum species associated with Siricid species that we see today is a very recent association. So selection for more successful associations there in more recent times than the diversification of those Siricid species, at least. So if we redraw this life cycle, I showed these in the beginning, these three different elements of the attack of the tree, of these stressed trees, the development of these larvae, and eventually emergence to continue this cycle. Then we now know that this is not a closed cycle, but that there is ample evidence for horizontal acquisition, both from other Siricid species, but also for exchange of genotypes amongst species amongst individuals of the same species, which explains what we see in these invasive populations. I'll skip over that. I pointed out the work that's been done to understand the acquisition of nutrition and the role of the fungus already. For the remainder of the talk, I want to focus on another symbiont within this symbiosis, another organism within the symbiosis, namely a parasitic nematode called Deladena Siricidicula. These nematodes feed specifically on these Amylostereum fungi. They don't feed on other fungi. They infect the eggs of the female Sirex noctilio, female Siricid wasps. And this female is not killed and her fitness is only marginally affected. She will take those infected eggs and lay them in a new tree where they can spread through the tree, find other uninfected larvae, and infect those, and ultimately infect their eggs again, and so stay within the cycle. So very tightly co-evolved system. This has been exploited, and famously in Australia and New Zealand, this has been developed for a biological control program, where you have this cycle in nature, where it either feeds on the fungus or infects the wasp, but you can isolate those nematodes then on the fungus in culture, multiply them by the billions, and reintroduce them into trees that might not be infected by the nematode, and so bring these populations under control. We've been studying the diversity of this system, similar to the Siricid and Amylostereum, and just to reflect on the early years of development, where 4,000 Siricid-affected logs have been collected from across Eurasian and Asian distribution of these organisms to collect these nematodes. And this is reflected at the top half of this image. So you see collections from many different parts of the world that was then brought to Australia and New Zealand, where it was studied, where there were initial releases of some of these, and where a particular strain, originally called the Sopron strain, later re-isolated and now called the Kimona strain, was used predominantly and spread within the biological control program. That particular strain was then taken and used in biological control in South America and in South Africa. So what you would expect, of course, is limited diversity. You've selected the strain, and that would be connected between these different regions. There's reason to reflect on the diversity of these nematode strains and the diversity of the populations that they're used to control. I've showed you earlier the diversity that we find within the Siricid populations, for example. Here we're expecting limited diversity in the biocontrol population, But there is old work that shows that some of these strains are able to parasitize some populations of the wasp and not others. So could that perhaps help explain some of the problems that we've had in applying this biocontrol agent across all of these different populations? Some of the additional reasons to think about the diversity within this system that has been shown in recent years is the fact it comes from the work of Anne Hayek and her lab. So Erin Morris's work here that's looking at different Amylostereum strains, so different fungal strains, that she's taking the nematode and looking at reproduction, and could show that there is vast differences and in the rate of reproduction of these nematodes on different Amylostereum strains. So thinking about diversity within this system for the sake of the biocontrol is certainly something that's worth considering. And that's why we have been pursuing that and trying to understand diversity within the biocontrol system, but also on a broader global scale. So here is work from Katrin Fitzer, microsatellites reflecting diversity of populations of the nematode that we could source from various parts of the world. And in yellow here, you see the diversity within our biocontrol systems. So as expected, very limited diversity there, and much more diversity that we can find in, for example, native populations. This lineage C comes from Europe. This lineage B was a separate lineage that was found in North America. So quite a bit of diversity there. That's more than what we have within the biocontrol system. Student Osmond Molinieni, who did his PhD on this work, looked at a surrogate for potential virulence within these systems and could show that there is a great diversity and potential virulence in these nematode populations. So more motivation to consider how this diversity in the system might affect biocontrol. And his work also supports the fact that if you take different nematode strains, put them on different fungal strains, then you get very dramatic differences in the rate at which they reproduce on different strains. So a very clear interaction between nematode and fungal strain diversity within this system. She also showed that if you took parental strains, so this is a lineage B from the US, and I can't remember what lineage this was from Chile, and we created a mixed culture of these, then we could increase the rate of reproduction of this. So we could use this diversity to increase something like reproduction, which potentially could influence success of biological control. So I'm moving to the last part of the talk and bringing all of this understanding of the biology and diversity in both the wasp, or in the wasp, the fungus, and the nematode, to a study that we recently did with Helen Urung and Angus Carnegie and other colleagues from Australia and New Zealand, and work that was done by Frihiru Setu and Katrin Fitzer and others, where we had a really fantastic collection with these colleagues. Nicholas Moraes from New Zealand also involved, of the Amylostereums, 185 strains, of Deladenas, 131 of those strains, and 500 collections of the wasp that we could then consider for diversity. And I'm going to now show you what we found in terms of diversity using both microsatellites and sequence data from these populations. And you're seeing here a representation of global populations, and I won't spend time going into all of this, but really reflecting the same very complex pattern of movement between different regions, admixture, a region such as South Africa, which is colored in yellow here, containing genotypes linked to many different parts of the world. The Australian population is here where you're seeing the big bubbles, because that was the focus of this study, so more isolates included from there. And interestingly, in this case, we also had access to historical samples, so preserved samples back to the 1950s. And that is what's represented in the orange circles here. So clearly this defined population and less diversity within this population, reflecting a good phytosanitary system, that has reduced the number of introductions in Australia relative to other parts of the world. The Amylostereum diversity is more than what we see in the vegetative compatibility group. So there's more genetic diversity if you measure it with something like microsatellites than when you measure it in a plate using vegetative compatibility groups. And what was interesting from these Australian populations is there was a fair amount of diversity, but these very dominant genotypes, which is good news for a biocontrol program because it means that there is a particular strain that you can focus on as the dominant strain within your environment and focus your research and development around something like that. But a good understanding of that fungal diversity that we then also get within that environment. The big surprise was, though, when we looked at the biocontrol nematode. We were expecting something that is very limited because by this one lineage called the Kimono strain, and that's represented in this lineage B in this blue circle, has been spread in the region for 50, 60 years very intensively. That was not the only of these lineages that we found there. We also found lineage C, and very surprisingly, a lineage D that we hadn't seen anywhere else in the world, and that was quite common within that. That is, I think, COX-1 data, and this is microsatellite data. So lineage D here, not rare within these populations, but actually quite common within these areas. So clearly something that has fitness, comparable fitness, or even advanced fitness to what is used in the biocontrol program. We've looked at the reproductive rate of these nematodes, or some of representatives of these different lineages on some of the prominent strains that's used in biocontrol and that is prominent in the environment. And here we maybe see a mechanism of why this lineage persists within this environment, where lineage D, which is colored in blue here, consistently reproduces faster than the lineage B on the various of these fungal strains that we put them, including the biocontrol, the fungal strain that's currently used in the biocontrol program. So a very clear target then to potentially improve the production, at least, of these nematodes, more diversity of the nematodes to target in the control program. And who knows, this remains to be shown, potentially ways to improve this program. I'm ending off by saying that this links to a concept that we've been exploring in the last while of thinking about not just integrated pest management systems for dealing with forestry pests, but really thinking about the diversity in our systems, both environmental diversity and biological diversity, and that we do need to think about precision in how we apply something like biological control, that putting a nematode into these populations, even if it is successfully sterilizing these populations, might not be an optimal strain that we're using, and that we need to understand both pest and in this case also this fungal diversity to develop more targeted biological control programs. And that is something that you can take to other forms of our management as well, where higher precision that is within our reach at the moment could be expected to give us better levels of control. So to conclude, these sericids were always thought, Siricida Amylostereum associations always thought to be highly specific and vertically transmitted, and it is so in most cases, but not always. We do have horizontal transfer not infrequently amongst them. So they're mostly asexually and vertically produced, but we also see the sexual reproduction being accessed and exchanged between the different wasp species as well. There's clearly specificity in the interaction between the nematodes and the fungus, and potentially between the wasp from older work. We haven't demonstrated that anytime recently. And understanding this diversity really offers us opportunities for selecting potentially more efficient biological control programs. So I've made this point in the previous slide, that understanding the diversity within these populations, biological diversity and abiotic diversity, really matters for how we're thinking about the management. With that, I'd like to thank you for your attention and thank many colleagues, both here at FABI and internationally, who've worked with us on these projects, and recognize Thorben Danke for these amazing images that I encourage you to go and have a look at on Twitter, that here of the ovipositor of Sirex juvencus. Jonathan, thank you very much for this opportunity.
Thank you very much, Bernard. That was awesome. Now we have time for questions. So either if you want to pop your question into the Q&A and we can read that off, or if you want to put up your hand, I can also allow you to turn on your microphone to ask it directly. While people are thinking of that, I have a couple questions of my own. So that swapping of fungal species or taking on horizontally of some of those new fungal species is fascinating. But if memory serves, you said at the beginning, you're not quite sure how it's getting into the mycangium, or did I misunderstand? Do you have ideas or theories as to how that's actually occurring?
Yeah, there's no direct evidence for how that happens, and it remains a bit of a mystery. As the larvae feed in the wood, they don't have internal structures to carry the fungi. They have an external structure in which they acquire these fungi. And the fascinating part of this biology is how they, in that non-sterile environment, can acquire that fungus so specifically. I guess it's like other closely associated insect fungal symbionts, a very pure culture of the surrounding fungal species into external pockets. So they have these external pockets where they carry it, and that is probably the mechanism. So if a wasp is to lay eggs in wood that is already infected by the fungus, and that larvae is to tunnel through that, it would acquire then the fungus that is in the surrounding wood. How it gets from those external pockets to an internal mycangial sac is not clearly known. There is speculation about wiggling of the ovipositor and the fragments from that external pocket moving up the ovipositor or growing up the ovipositor into that mycangial sac. But yeah, direct observation of that has not been done.
And where they're picking up fungi that is new to them, have you been able to see if that is positive for the larvae? Are they growing faster? Are they growing bigger? Are they, is there any fitness advantage to that for them?
Yeah, that we haven't demonstrated that. I mean, when I look at these phylogenetic trees and this sort of recent diversity of Amylostereum species associated with Siricide species that have a much deeper evolutionary history, then clearly that must offer an opportunity for selection all the time, and that fitness, potentially, growth and development of those larvae could be one of the factors that get selected. Could explain that picture of the distribution of that Amylostereum diversity that we see amongst current Siricides. But yeah, no, I don't think anybody has demonstrated such a direct effect after such a transmission.
From our participants, does anybody have any questions that they would like to ask?
While we wait, I'd say that would be quite a cool experiment. These horizontal acquisitions, we've always only showed indirectly. So we can see that Sirex nigricornis in the US carries a fungus that it shouldn't be carrying from a wasp that's just been invaded. So it's shown it indirectly, but we haven't actually inoculated a piece of wood with genotype A and then put the Siricides on it. So that would be a cool experiment. The trouble and the difficulty in working with the system is they have year-long life cycles, though. So it's it doesn't fit into the typical postgraduate project and really requires a lot of patience to work with.
Fair enough. Yeah, we always complain if we have a couple of months experiment that falls over flat. A year-long experiment, that'd be really awful. I have another question related to the nematodes, actually. So if I am understanding correctly, the nematodes are feeding on the fungi, correct? So yes. Then how are they controlling the insect itself?
So they actually have two morphological forms. one morphological form that is during which it feeds on the fungus, and you can maintain it in that form indefinitely on a culture. You can maintain it indefinitely. In the presence of the wasp, the eggs develop, or the young nematodes develop into a second morphological form that penetrates the larvae and develops inside the hemocell of the larvae, eventually reproduces inside it, and those young juveniles will migrate to the eggs of the developing larvae, penetrate the eggs, and sterilize those eggs. The wasp still emerges, and she's just filled with eggs that have nematodes in them and are not going to produce wasp offspring. So she spreads the nematode to the next tree. So it's a very nice classical biological control system in that sense.
Yes, I'm going to have to use that in my biosecurity class now. Carl has a question. Would seasonality and weather patterns, example, wet years versus dry years, affect that fungal infection status?
Probably, yes and no, in the sense that it probably has an effect on the number of trees that are susceptible to this infection. So a healthy, vigorous growing tree typically is not going to succumb to the infection. So even though the wasp introduces the fungus and the phytotoxic mucus, that fungus is not going to persist, and ultimately rot the tree. The tree is able to overcome that infection. So that would be the reason why the wasp also doesn't introduce her eggs at that point. She recognizes that it's not suitable for the development. So in dry years, or in particularly hot years, there would be more trees that are stressed, and they are simply more susceptible to both the fungal infection and ultimately for the larvae to develop. You typically get higher numbers of attacks under those conditions. So there's also more of these infections on that particular tree, further stresses it, and that gives a hold to the tree. So yes, there's more opportunity for the fungus, but the effect really is a bit indirect.
Great. Okay, last question for me, and then potentially you're off the hook unless somebody else has something. Going back to the nematodes, then you said that lineage D you'd never seen anywhere else in the world, only here in Australia. So if it's endemic here, but the wasp and fungi are introduced, should we be looking for some of these nematodes in other fungi, or no?
No, let me explain that. So it was a surprise to us. So I didn't explain that clear enough. So when you use this in your class, I need to do better on that. The nematodes were introduced from this broad collection across its native distribution in Eurasia, from Asia, from Europe, brought to Australia. And while the biocontrol program was developed, there were releases done in the field. Some of these releases must have persisted. What we're seeing in other parts of the world is now selected strains that have been brought there and used in biocontrol programs. And the nematode is not native to Australia. The only explanation for that is that it is some of the early original releases that have persisted all these years, despite the fact that those populations have been inundated with lineage B, which is used in the biocontrol program. So one assumes that there must be some fitness advantage that has allowed it to be dominant even in some of those populations. And this last experiment I showed, where lineage D seemed to reproduce faster on most of the fungi we can present it with than lineage B, might be the mechanism. So it just produces higher numbers within the tree, can find more larvae and persist more effectively in the environment. But that potentially gives us a really clear path to now select from this diversity strains that might be better to use in our biocontrol programs. And that is something that remains to be demonstrated, but certainly seems to have potential.
Great. David Guest has also a question, says, thanks, Bernard, fascinating. What is the potential of the wasp phytotoxin as an herbicide?
That is a fascinating question, and something that I know some colleagues have been considering exploring. It's a small protein that's produced, and I can't remember the name is actually noctilicin. It's called noctilicin. So there's actually publications on that. But yeah, and it's been shown that, you know, you can isolate that and introduce it and have this wilting effect on pine. So that idea has mulled around people's heads, but nobody has directly explored that. Certainly would, in areas where pines are invasive, was the thought, would have really fascinating potential. Difficulty, I think, in that, in some of the discussions were thinking about how one might actually apply that, you know, introduce, because you'd have to somehow introduce it into the tree, and doing that on large scale in individual trees would be pretty difficult.
Peter Buchanan asks, what was the expected source of Sirex and Amylostereum for New Zealand, and was that the same source for Australia?
Yeah, I think that if I recall correctly, predominantly looks like a European source. We don't see the connection between New Zealand and Australia very clear. So those two populations more or less looks like one population. They are less diverse than what we see in some other parts of the world. So clearly have not suffered from this multiple introductions from different regions and the subsequent admixture as we have in South America and in South Africa. Yeah, so those two populations quite closely related to each other, clearly connected. So that historical move from New Zealand through Tasmania to Australia, quite evident in the genetics as well.
Great.
I mean, one side comment there. I mean, what was fascinating to see in nematode diversity, for example, in places like South America, is links of introductions from New Zealand into South America, for example, Australia, New Zealand area, into South America, into Africa. So one can see the movement from that region to other invaded populations, but not towards them.
Fascinating. Well, thank you very much, Bernard, for getting up early, for talking to us. Thank you for a great presentation and such great answers.
Jonathan, yeah, thank you again for the opportunity. Gets my day off to an exciting start, and I wish you all a good afternoon and evening, and hope to see many of you again soon at the conferences.
That sounds great. Thanks very much, and bye, everybody.
Goodbye.